Hemostasis. This is where we are, it's the process of stopping bleeding and returning everything back to normal. And so it's all these events that prevent excessive blood loss. So the first thing that happens is a vascular spasm where the vessels will constrict to try to stop the blood flow through them. And then a platelet plug will form, and then that will coagulate and the blood will clot. So we're going to go through those events. Ooh. So here, this platelet plug formation, we have the platelets that will come in and they attach to the collagen that's in your skin and in your tissue. They'll attach there, and then they will attach to other platelets. And all these platelets will come and bind together and plug up that injury site, wherever it's that platelet plug. And then the coagulation will occur where prothrombinase will activate the prothrombin into thrombin. So prothrombinase will convert the prothrombin into thrombin, and it also converts the fibrinogen to fibrin. So these are the active forms that will actually go and start making the clot. So all these, the prothrombin and fibrinogen, they are all proteins that are just found in the plasma, and they're floating around all the time until they're activated. So they are circulating in the inactive state until some tissues are damaged, and then when the damaged tissues and platelets start producing these chemicals that activate them, then they become active and will form clots. So the result is a blood clot, and what happens is the fibrin makes this thread-like well, if you look at that picture, it's the yellow parts, and it looks kind of like gauze, and it's going to trap all the red blood cells, platelets, and fluid. And that's actually what a clot is.
I think it looks like pus flavored cotton.
Yum.
Sounds good.
Yeah. So this is just a short diagram showing the formation of a clot. We have an injury, so the connective tissue is exposed and the chemicals are released, which activate the clotting factors, like the prothrombinase, and the prothrombinase then will activate the prothrombin and fibrinogen to form the clot. Back a slide. So you can see there's a lot of different steps that it goes through. That way you don't, that's to help prevent your body from just producing clots on a regular basis when you don't need it. So it goes through several different steps to try to keep your body to create clots only when needed. It has to go through all those steps.
So why do unhealthy people get blood clots that break loose and go to their heart?
There's different reasons why people produce blood clots.
Oh.
That's not just, yeah. There's something has gone wrong in that step. And when you have hemophilia, that means you don't clot, and there can be a problem with the thrombin and the prothrombin there, or it can be a problem with the fibrinogen. Like there's just... So that is another, because there's so many steps, it helps prevent your body from producing clots a lot, but there's a lot of different steps that can go wrong too to prevent formation of a clot. So to control clot formation, you have anticoagulants would prevent the coagulation factors from initiating clot formation, like heparin, or if you're any other blood thinner, is an anticoagulant. Coagulation occurs when coagulation factor concentration exceeds a given threshold, and at a site of injury, a threshold is exceeded, and that's when those factors are activated. So some of the anticoagulants are antithrombin, which is produced by the liver, and will slowly inactivate the thrombin, and the thrombin's what's activating the fibrinogen. So if you inactivate the thrombin, it's not going to activate activate the fibrinogen, so it's going to stop clot formation. And then heparin is produced by basophils and endothelial cells, which will just increase the effectiveness of the antithrombin. What does the suffix lysis mean?
To split.
To split, to break apart. So if we have fibrinolysis, what do you think we're breaking apart? The fibrin, which is what's creating the clot. So breaking down the clot, when it's done, so the thrombin and tissue plasminogen activator activates the plasminogen into plasmin, and that would break down the fibrin.
So the plasmin is what breaks down the clots.
Mm-hmm. Yep. So plasminogen is floating around all the time in its inactive state until it gets activated into plasmin. Clot retraction, the fibrin has some actin and myosin in the platelets also, and they start to contract and pull everything in together. So serum is then squeezed out of the clot, the liquid part of the clot squeezed out, pulls the edges of the damaged blood vessel together also as it's contracting. The clot is then dissolved by the plasmin, which hydrolyzes the fibrin. Antithrombin will help activate the plasminogen. Okay, blood groups. Everyone's probably heard of, like, I'm A positive or A negative, whatever blood you are. So there's the ABO groups, which are either A, B, or O blood type, but the positive and negative is your Rh factor, whether or not you have an Rh factor on your cells. So these are very important when it comes to transfusions, because if you transfer blood from one type to another, you could get a reaction, transfusion reaction it's called, where it will start coagulating the blood that's incorrect type. An infusion is the introduction of a fluid other than blood, so like just plasma or an IV. Those are all infusions. Infusing a gas. Is that what you said? Oh, potassium, yes. A gas. Early transfusions were unsuccessful because of those transfusion reactions. They didn't know about the blood type, so they just had very bad luck using those transfusions. And so there's antigens on the surface of a red blood cell. These markers, they're called antigens on the surface of the blood cell. And then there's antibodies that are freely floating throughout your body, throughout the blood. Those antibodies don't match the antigen. In your body, the antigen and antibody do not match. So there's two different types of antigens, two different types of antibodies that you can have. They do not match in your blood. Right. If you're given blood that is the wrong type and it has the antigen that matches the antibody, those antibodies cling to it, and that's what forms clots and problems. So that's why you need to know what blood type you are. So this is just, I mean, it's a simplified pictures. The markers don't look like this and everything, but same idea. So if you have type A blood, that means your red blood cells have antigen A on them. That means you have antibody B floating around in your blood. They don't match there. If you have type B blood, you have antigen B on your blood, on your red blood cells. Antibody A is floating around, the anti-A antibody, I guess. If you have type AB, that means you have both the A and B markers on your red blood cells, but you don't have any antibodies in your blood. So you can basically receive any type of blood except for O. In smaller amounts. Wait, I take that back. You have to get only AB blood. That's right. O, you have neither antigen A or B. And actually, I do take, because the type AB, you can actually receive any blood. That's right. Because the antibodies that are in the blood that you receive, there's so little of it. It's not like you have a bunch of it. Like your blood system, you don't get that much blood. So you can take some medications to kind of take that back. Because that is the universal receiver, where type O, they call it the universal donor. Because the red blood cell of the type O has none of the antigens, so it can go into any of those blood types and it won't have a reaction. Now, it does have both antibodies, so that little bit of antibodies that you get with the transfusion may have a small reaction, but not like if you did the other way around. So there's still some risk if you don't get the same exact type, but that's why they call the type O the universal donor. Even if a type A person gets type O blood as a transfusion, there is a small risk. There is still a risk that you can have a transfusion reaction, but it's better you get type O than type B that way. So this is just showing how that can form that reaction, agglutination reaction, if you get the wrong type of blood. So the ABO antigens appear on the surface of the red blood cells. So type A blood has type A antigens, anti-B antibodies. 41% of people in the U.S. have type A blood. Type B blood, you have type B antigens and anti-A antibodies, and only 9% of the population has type B blood. So if you have type B blood, I'm sure they really want you to donate. Type O blood has neither A or B antigens and both anti-A and anti-B antibodies. And 41% of the population is type O. Actually, the highest percentage is type O. And then type AB blood have both A and B antigens, but none of the antibodies, and 3% of the population. So tomorrow when we do our blood typing lab, that's what we're going to be looking at. We're not using real blood, but it's going to be the same idea. You'll have different types of blood, and you're going to be putting anti-B antibodies and anti-A antibodies in the blood and determine what type of blood it is. Whether it's not real blood. It's synthetic. But that's the same when they test your blood to see what kind of blood you have, that's how they do it. Same process. Okay, switch it. Yeah? Not yet. I don't know. Don't they kind of process your blood after you donate it before they use it? I would think they would, or do they just test it? I know they test it, but do they do anything? Like, I'm wondering if they can inactivate the antibodies or something, so that way you're just getting red blood cells. I don't know. I'm not for sure. They can't inactivate your antibodies. That's why the antibodies that are in your system is more important, like causing those reactions. But I don't. So it's suggested that the antibodies are present because of an exposure to an A and B antigens on bacteria and food. Because when you have infections, that's what your body is producing is antibodies, and they attach to foreign cells or foreign particles, and that's how your body knows to attack those cells. And so one of the theories is that because of bacteria in the food that we eat, you're exposed to those antigens on those cells, so your body produces certain antibodies to fight that. Type O is called the universal donor, but you can cause a transfusion reaction because of the antibodies. So you just have a higher risk than just getting the correct type of blood. So type O blood is given to a person with another blood type only in life or death conditions where they absolutely need a transfusion and there is no blood in their type available. Okay, the Rh blood group was first studied in rhesus monkeys. So you have Rh positive and Rh negative. So if you're Rh positive, you have the Rh antigens present on this red blood cell. If you're Rh negative, you do not have those antigens. So if you are A positive, that means you have both the A antigens on the red blood cells and you have the Rh antigens on your red blood cell. If you're A negative, you would have the A antigen and no Rh antigens. And normally you do not have antibodies associated with those in your system unless an Rh negative person, so you don't have those Rh antigens on the red blood cell, is exposed to an Rh positive red blood cell. Then your body will develop those antibodies, and the next time you're exposed, it's going to coagulate that blood. And this is very important and dangerous with mothers and their newborns. If the fetus is Rh positive and the mother is negative, and it's called the hemolytic disease of the newborn. So the mother is negative. The mother would not have those Rh factors, but the fetus does. Late in pregnancies, the Rh antigens from the fetus can cross the placenta, and since the mother then is exposed to Rh factors, the mother would develop the antibodies. And it's not a problem during that pregnancy, but the second pregnancy, if again the fetus is positive and the mother has negative, since the blood from the mother goes to the fetus during pregnancy, it can cause, since it has now the antibodies, it can cause the red blood cells of the fetus to coagulate, since the mother's antibodies are now going to the fetus that has the Rh factors. No, the fetus doesn't have the antibodies. The mother would already, in the second pregnancy, the mother would already have the antibodies. So it's going to, if it comes into contact with positive blood, again, then it's going to coagulate that blood. And since if the second fetus is positive, the blood from the mother is going to the fetus and will coagulate the fetus's blood. So if you know, usually they'll test blood and they know blood types, and so they'll know if in the first pregnancy if you were exposed or whatever, and so they'll know the second pregnancy they need to take precautions, and so they can give you RhoGAM, it's called, and so it contains antibodies against the Rh antigens, so they can prevent the mother from being sensitized to it and can prevent that from happening. So they can prevent that as long as they know the blood types. I'm pretty sure erythroblastosis fetalis is another name for that hemolytic disease of the newborn. It's just showing a picture of what happens here. Okay, so to diagnose what blood type you have, they use a type and cross-match test, and they have antibody serums and solutions, so they just take a sample of your blood and test it with those serums to see whether or not it coagulates. So if you have type A blood, well, let's say if your blood, when they add anti-A antibodies to it and it coagulates, what type of blood would you have? Right, it would be A. If you have blood and they add anti-A antibodies to it and it coagulates, so it clumps up, what type of blood do you have? A, right? Because you're adding the anti-A antibodies.
Wouldn't they have to check it twice just to make sure you're not A?
Yeah, they would test them both. So then they would test the type B too, and if type B does not coagulate, that means you would have type A. If it would coagulate also and both of them coagulate, that means you do have type AB blood. If neither of them coagulate, what kind of blood would you have? Yeah. Some other tests that they can do are red blood counts, so they can measure the amount of red blood cells per microliter, a hemoglobin measurement, grams of hemoglobin per 100 milliliters of blood. Did someone say in here that they had theirs tested and theirs was like at 10? Maybe that was
Mine was great.
Amy. I think Amy Hamburger yesterday said that after she donated blood, she got anemic and her hemoglobin measurement was like a 10.
Not bad.
Well, normal would be 12 to 6, so she had 10, or 12 to 16. Yeah. A hematocrit measurement is the percent of blood that's a red blood cell, so they'll take a little sample of your blood, put it in a vial, put it through a centrifuge. You guys know what a centrifuge is?
Yeah.
A thing that spins really, really fast, and then so it makes the heavy stuff come to the outside, because usually the vials are kind of angled like this, so all the heavy stuff will go to the bottom of the vial, and then they can measure percentages of red blood cells.
Will you get one from Church?
No.
Urine.
Did you use real urine, or did you use fake, synthetic? In biochemistry in college, we actually tested our own urine for vitamin C.
How did you, was it a taste test?
No, you had to go in a cup, and then you brought it back to the lab.
That'd be difficult.
White blood cell count. If you have lower white blood cell, it's leukopenia, and then higher leukocytosis. But if you have the cancer of the white blood cells, is the leukemia. And, yeah. So here's the hematocrit, where they would spin it, and you could see the formed elements, the red blood cells, are coming to the bottom. Differential white blood count determines the percentage of the 5 types of blood cells. It gives us our normal range there of each of those. You can do clotting. You can do a platelet count, where they would actually measure the amount of platelets you have per microliter of blood. Or you can do a prothrombin time measurement, which measures how long it takes for the blood to start clotting. Should take 9 to 12 seconds. And you can test it by adding thromboplastin to the plasma. Now there is another way, and I don't know, maybe they don't do this anymore, but my aunt had a clot. She died from blood clots and had a clotting disorder, and so before they did took our tonsils out when I was little, they had to test how well we clot just because that was it was going to be a major incision and stuff. And so what they did, and I still have scars from it, they took a little had two little razors and they clipped your skin. So I have two little marks here where they opened up your skin, and then they took like a filter paper, the circle filter papers, and they just every 2 seconds they would dab it on the blood, and then they time how long it would clot. So I don't know if they do that anymore or not, but I don't know. Is that it?